Okay, so our first sort of warm-up poll question is what best describes your perspective in the consortium? So industry, government, academia, nonprofit, or other?
We're also, I do want to say, and people have mentioned with the government shutdown, we've lost about a third of our participants today. So
Yeah,
it might not be shocking that we have a 60% industry today. And we might have government from other jurisdictions, which is awesome that we still do have that participation. But I think it might be slightly skewed today just because of the shutdown.
yeah. As I was putting it together, I was thinking of that. Great. So still pretty good representation given that the US government is currently shut down and many people weren't able to join. So we'll go to the next sort of warm-up question. So which of the following best describes your level of engagement with the Botanical Safety Consortium? So the options are active member of one or more working groups, a BSC member but not currently active in a specific working group, not a member but regularly follow BSC activities or updates. So I know some people come to our annual meetings almost every year, and then new to the BSC / first time attending a annual meeting. So it's nice to see lots of our working group members were able to join. I know we've been reminding you all about the meeting for probably 4 or 5 months now. And a good number of non-members, but who frequently follow the BSC activities, which is nice to see. It makes me feel like our outreach is validated. Great. All right, so now that we've done the warm-up questions, everyone should be a little bit more familiar with the Menti platform. We're going to go into some open answer questions. So the first one is, which areas should the BSC prioritize for further work? More botanicals?
Yeah, like Julie said, this can be open-ended, but if you're like, oh, we really need to test 50 botanicals, that's it. Or if we need a renal toxicity group, that we don't have one of those, or
Yeah,
inhalation, this is where we really would like to hear from you all.
so botanical drug combinations, guidance on determining botanical constituents. I know one topic that's come up a lot in our working groups is, and I think was discussed in almost all of the presentations today, was like lot to lot comparisons and seeing how the different lots respond in the assays. So I'm going to hit space. Theoretically, AI will magically start grouping our answers.
This will show if we still need me and Julie or if we can just AI everything.
Yeah. Some more botanicals. We have about 8 responses. In vitro, which is a pretty broad category Menti, but if we click on them, so it seems like sort of IV, IVE, so the in vitro to in vivo extrapolation. Constituents. Oh, we have more responses that weren't categorized. So if I hit space again, no, it doesn't want to redo it.
Maybe we
I
are
was
still
too
making
early.
moves.
Yeah. Potential use of botanicals in cosmetics,
Hmm.
mechanistic insights. Great.
I
And we'll
like the
be able to,
combo botanical one. That was interesting. That's something we've talked about, like multiple
mm,
botanicals. That's like a whole nother level of complexity. Well, that's cool.
yeah. Yeah, and I know if you look at some of the botanical products available on the market, there'll be like a combination of 10 different botanicals to look at. And Connie and I will have all of these individual results, so they'll help guide next steps potentially for the consortium.
Yeah.
All right. So, oh, you can vote if you want. I don't know if we need that. Uh, so we'll just go to the next slide. So one question we had is, where could cross-working group efforts add the most value? So I know Shane mentioned potentially like cross working group efforts between the neurotoxicity working group and the cardiotoxicity working groups due to some similarities in the signaling. Uh, so we were wondering what other cross working group efforts could be beneficial. Yeah, add me with pretty much all other groups, especially with these in vitro methods. And that's related, I think, to the IVIVE as well. Genotoxicity and hepatotoxicity, analytical with all others.
Yeah, one thing we didn't really talk about today, but it's come up in probably every work group over the years is, for some of the assays, could we almost keep the cells or the test system and then quantify key constituents that are actually in there to see what's getting in? It's not something we've done so far. I think it would be really cool, but a little bit tricky. I mean, but then if we already know what markers to look for, maybe we could do it in select cases.
Yeah,
Ooh, metabolomics. Folks won't know I also manage our omics work, so I get excited about that.
let's see. So core battery, so comparison of neuro, cardiac, respiratory. We don't currently have a respiratory group, but that could be something to expand to. IVIVE.
Yeah, some of these are like gene tox start, if something is potentially causing genotoxicity, you think it might disrupt how the cell is developing and going. So that might be really cool. Probably gene tox
Yeah,
in a lot of these categories too.
yeah, much like ADME in a lot of the categories.
Very cool.
Yeah. See if we have any trends from the about 20 answers we've gotten so far.
Add me, gene
Yep.
tox,
Transcriptomics, IVIVE,
analytical.
metabolics. Cool. This will be really helpful. I'm gonna skip through the voting again. So this one I believe should be a word cloud. So it is which technical challenge most limits the practical use of NAMs for botanical extracts? So it could be, yes, solubility. I know this is one that's come up in quite a few of our working groups. The word clouds are kind of fun. I know another concern was potential matrix effects or binding of active constituents to the actual test systems themselves. That's always a concern for some of the new approach methods that are done in the plastic well plates.
Yeah, I like the lot to lot comments as well, because you all probably have heard, except for a few pilots that we did on the analytical side, not the assay side, we really just chose one lot to begin with. So, and
Hmm.
then there were questions too of like, we got the plant. Our colleagues at University of Mississippi helped us create an extract. But if we did either of those steps again, would we get something slightly different? And the answer is probably. And then how much would that affect the bioassay? So
Yeah.
that is a question of, we have a pretty well characterized library of constituents. Of course, we would all love to go deeper and know everything in there. But yeah, if we did any of this again, would we get the same? So that's why we're also very trying to be very careful that we're assay focused and not, trying to walk away with a conclusion about milk thistle.
Yeah. Thanks, Connie. I think there's a couple about oh, it keeps moving as I try to focus in on things like concentration determination and identification. I saw one that's too much data, which I agree is overwhelming at times,
But that's true if
but
you're
good to
trying to make
have.
a decision. That's the thing. We're trying to put these together and put these pieces together. But sometimes it can be hard or conflicting data.
Yeah, if you run a battery of 20 assays, what does it take to find a negative?
Yeah. I also appreciate human relevance is pretty big. I don't think that's specific for botanicals. It's a NAMS issue generally, but that doesn't mean we just ignore it, right? So that is something we're trying to, again, put these pieces together, think about ADME, think about concentrations, but what's actually getting to a human? And, is it always a healthy human that has the issues? Probably not.
Mm-hmm. Right,
Lots of
and it seems like,
nuances.
yeah, Seems like we've stabilized, but definitely human relevance, solubility, mixtures, chemical complexity, and standardization seem to be the big ones. And it seems like even some of the smaller font size are also tied into those bigger concerns. Thanks. All right. And then this one's more like a future directions for the BSC. So it's a ranking question. And so the request is that you rank the following areas in order of priority for follow-up and next steps. So one would be the highest priority. I'm going to guess based on previous answers, ADME will be ranked fairly highly. Yeah,
Ooh, we might have a duel though.
I know. And lots of lot variability is really coming up in the ranking.
And one thing we talked about it in two of the presentations, but for neuro, for I think about 8 constituents for the MEAs, we compared individual constituents with the mixture in the same lab, same setup. And then we didn't spend a lot of time talking about today, but I think it's a really cool study we did with Axiom. We ran 80 of our constituents across the botanicals and compared that to the mixtures. So I think there's a lot of really cool things we've generated data on that we need to look at. But that's something we could potentially look at for other assays. For some that maybe aren't validated, sometimes it helps for that understanding, benchmarking single constituents with the mixture effects too, which might
Hmm.
give us insight of, are these usable for botanicals as complex mixtures? The tricky thing is, as we talked about, it's not always one constituent driving it.
Yeah. All right. So I do feel validated that ADME ended up in number one, but it was quite a close race between that and the extracts versus isolated constituents. Give it seems to have stabilized. So I will move on to the next. Oh, we had one more. All right, I'll move on to the next question. So this is what's one key question or uncertainty that you would like answered before next year's meeting? No guarantees we will be able to answer it, but it would be helpful to know what you all view as a key priority for the next year or an uncertainty that you identified when listening to today's talks. Yeah, and I think one thing with the ADME is because we largely have in silico information right now, it's for those individual constituents. And so comparing that back to the whole mixtures can be complicated, but maybe that's an area where incorporating individual constituent information would be helpful, like in the actual assays.
I think we can promise on the more dermal data as some of those are
Yeah,
getting going. So that's one I think we can at least say they're initiated by the time we meet next year.
yeah. Identification of which assays are most valuable to screen.
Ooh, AI modeling. That's of course the buzzword, but if people have expertise with that, if we could throw a bunch of data and it could put things together, I mean, as a pilot, I think a lot of these would be, that could be cool. So if someone has
Yeah.
ideas on that,
And I think generally there's a lot of interest in like using model like AI to integrate data from either different constituents and try to model like a mixture effect or from integrating the different toxicity assays themselves.
yeah, validate token incorporation into assessment framework.
Gut microbiota.
We have, I mean, in the tiniest bit, thought about that with like gene tox in silico that you wouldn't necessarily throw just because it's not getting it absorbed. You still do have to
Hmm.
worry about like gut tox. That's not really microbiota, but it's something of thinking in the gut. And then even if you have sugar molecules, the microbes there might be able to break off the sugar molecules, so you might have more active forms of the constituents.
Hmm.
So maybe we could do more than that in the next year, but that is one thing that just came to mind when I saw that.
Yeah.
Ooh, nephrotoxicity. We wrote one paper. We got asked to write a paper that we had a really excellent postdoc at NIHS led. And so we have a review paper on nephrotoxicity where we looked at assays and botanicals, but we haven't heard a ton of interest for that as a full group, but maybe now's the time.
Yeah.
Lots of having public, yes, that is one we really want to have so people can play these data so data will be publicly available. Cool.
Well, gastrointestinal metabolism. In a
I
group.
definitely think some of these, like, yes, next year we will have some of these. It's like, have we considered them?
Yeah. So data integration, toxicity predictions, addressing human relevance.
I think that's really cool with the what Catherine was starting to look at and some of the dose information.
Hmm. Regulatory implementation.
We're not regulators, so that's up to y'all. But it is like, having these conversations with FDA, what's useful for them, what they want to see, and non-FDA, right? EFSA, Health Canada, others. So, we're trying to make the science available. That's why I threw my hands up at that.
Mhm.
But, to be a little bit funny, but really we're trying to lay what's actually known about these. How can you use these tools? Do you need to make any changes? But then that piece, like we're happy, to speak about what we've learned with folks.
Yeah, we're gonna finish. All right, and so then this one is more just like general feedback about either the annual meeting, the content of the annual meeting, or the BSC in general. Thank you. I'm glad that the first comment was very positive. All right, for this one, I don't know if we all need to review the general feedback, but I'm going to leave the question open and just stop sharing my screen. So that way you all have an avenue to provide your feedback. And then if there's any additional discussion uh, folks want to have, you can raise it in the Q&A or perhaps raise your hand.
Thanks, Julie. I think that gave us a really nice snapshot of, what we've heard today, what people would like to see in the future. I don't see any questions in the chat. I'm happy if anyone on the panel had any kind of cross-resonating thoughts or things they would really like to focus on or to see, let's say by this time next year. So maybe open up to the panel if anyone has any thoughts. I know it's lunchtime too. That's the tricky part. And late for some. Catherine might be doing her Guy Fawkes celebration already.
Oh yeah, and hopefully Shane went to bed.
Yeah.
Yeah, not quite yet, but fireworks are starting to go off, so I will stay on mute.
Bill said, I would like to see studies include equivalent human doses to see relevance. Yeah, that's a really fair point. And when we started, we really did based on limit of solubility and then tried for as many as we could to have these long ranges. I think this is where others that know more about IVIP can say more, but maybe there's some where they're totally not getting in on the constituent level. We can at least say, throw those ones out. Would that change our call? Or is it something that, okay, we saw toxicity in the assay, but that's really not something we expect to see? So I think there's some things we can do with the data we have, but then in the future, yeah, is that something we do a little bit backwards, projecting on what's a human taking and then put that in there? Yeah, so Christine, come on. Go ahead, Christine.
Connie, that raises a thought in comparing our NAMs, our in vitro assays, with the human exposure situation, in that I bet most humans that are taking one or more botanicals are taking them on a more chronic basis, whereas what we're doing in vitro is an acute exposure
Yeah.
situation. So that adds another layer of complication to being able to correlate the assay dose levels with what humans will experience.
That's a really good point, Christine. I know, we mentioned very, very briefly for some of the cardio talks that some of the gentlemen there went up to more like 7 days. But is that even chronic, even though it's still in vitro, but some of these are very short exposures. So I think that's a good point. We're almost in this phase of can we do an acute screen but that's not always going to capture some of the effects. Some of them you have to get to a certain level inside to see effects. So I think that's really worth raising as a limitation so far and something we can think about. Good question. Okay. Well, I know it's been a long day. We went through a whirlwind of data. I hope we can continue to share, exciting papers and things with you all. And as the data sets are made available, make sure people know and ideas, even when we make a data set public, there's still other analyses we can do with them. So we're going to do our best and within the groups decide how to package it. But I really encourage folks if they have ideas for things we can do with the data we generated or things we could do moving forward to let me and Julie know. I think with that, we can wrap up a little bit early. I really appreciate everyone's time. I appreciate our panel so much for walking through the different work. And I really just thank you all for your attention. And we will send the recording out in the following days. All right. Thanks, everybody. Take care.
Thanks, everyone.
Okay, thank you. Bye-bye.
Bye, everyone.